Good morning. It's a pleasure to be here. I recognize we are rapidly, and more rapidly than I thought, approaching the lunch hour, so I will do my best to occupy your attention for the next few minutes. I'd like to thank the Alliance, the meeting organizers, and the sponsors for allowing us to participate. And I hope over the next few minutes I'll be able to share what we think is a very exciting story about AlloCure. So we are a company that is focused on kidney health. We've heard about a lot of other organs and organ systems that are the focus of attention of companies at this meeting, but our clear attention is focused on the kidney. Next slide. We are a clinical stage, venture-backed biotechnology company, as I said, focused on kidney disease. Specifically, we are developing ACT-607, which is a mesenchymal stem cell therapy, which is being directed towards an area of unmet medical need in kidney medicine known as acute kidney injury, or AKI. ACT-607 has been awarded fast-track designation by the FDA, and we currently are in the midst of a large phase 2 proof-of-principle clinical trial, which we call ACT-AKI, which will enroll 200 subjects. The readout for that trial will occur as planned in the third quarter of 2014. So a little bit about acute kidney injury, and I pulled this cartoon from a review article that was published last month in one of the nephrology journals. This cartoon serves to illustrate a few key points about kidney injury, the disease, and the consequences that society faces as a result of this. First of all, acute kidney injury is all too common and occurs in patients who are unlucky enough to be in the hospital for other reasons. It affects in the neighborhood of 1 to 2 million Americans every year, with similar numbers in the developed world in other countries. When acute kidney injury develops, since it's happening in patients who are otherwise hospitalized, often in the ICU, it leads to extension of their stay in the hospital, often in the ICU. It leads to adverse outcomes, including the need for short and often long-term dialysis support, markedly increased in-hospital mortality, and the attendant risk that they are predisposed to long-term kidney dysfunction, chronic kidney disease, and the potential progression to end-stage kidney disease when long-term dialysis and transplantation is required. So when we look at that and then take a perspective to think about the health economic ramifications, they're extraordinary. And unfortunately, there are no approved therapies for this all-too-grievous condition. So as I mentioned, acute kidney injury most commonly occurs in patients who are in the hospital, and it can occur in a variety of settings. And the way that the equation kind of builds is that you have a patient who has underlying comorbidities. They may be elderly, they may have high blood pressure, they may have diabetes, they may have some underlying chronic kidney disease. And on top of that, they are then exposed to certain insults as we attend to all their other clinical conditions. For example, they may go to the operating room for CABG surgery, coronary artery bypass, graft, bypass. They may have valve surgery conducted on their heart, and when they're in the operating room, they undergo they're placed on cardiopulmonary bypass. That can contribute to the risk for the development of acute kidney injury. Similarly, they may have sepsis and hypotension and be hospitalized in an intensive care unit where they're administered nephrotoxic or kidney toxic antibiotics. That can be another setup for the development of acute kidney injury. Or finally, they may be prescribed contrast imaging agents in advance of going to a radiology suite for a CAT scan or perhaps the cardiac catheterization lab for a cardiac cath procedure. Those ionic contrast agents are nephrotoxic, can damage the kidney. And so these are different settings in the hospital where patients are predisposed to the development of acute kidney injury. As I mentioned, when it occurs, the complications can be quite serious, both from a health economic perspective and also from an individual patient's perspective and their outcomes. So what is the approach to think about ameliorating acute kidney injury with a regenerative medicine strategy? And for AlloCure, that is to leverage the opportunity to utilize mesenchymal stem cells to instruct damaged kidney tissue to undergo a reparative process as opposed to a fibrotic process. So AC-607 is derived from the bone marrow of young, healthy donors. We have a mature and modern manufacturing process that allows us to scale up the mesenchymal stem cells and keep them stored frozen at clinical sites around the country. We are comforted by the fact that mesenchymal stem cells, as we all know, have been utilized in clinical trials in a variety of settings and now have been characterized by product approvals by regional health authorities around the world. Importantly for us, AC-607 is immune privileged, and as an allogeneic therapy, there's no need for specific tissue typing or blood typing of the recipient. That is, we can think about this as a rather traditional off-the-shelf biologic form of therapy for patients who are in need. So let's talk a little bit about how we think this is relevant in this form of kidney disease. So we know that in the kidney, when there's acute organ injury, there's upregulation of a specific chemokine, stromal-derived factor 1. MSCs have on their cell surface the cognate receptor for SCF1, which is CXCR4, and that allows these cells to migrate to the site of injury based on that chemokine gradient, where they take up residence for a transient period of time. And we very much think of this as a paradigm of local, transient polypharmacy effect. That is, when the MSCs take up residence within the injured organ, they secrete a myriad of relevant paracrine effectors that instruct that local damaged kidney tissue to divide and survive rather than undergoing apoptosis and ultimate organ fibrosis. So the biologic story that supports this begins with recognition that in the setting of acute kidney injury, there are some things happening simultaneously that teleologically would seem to encourage organ repair. So first, we know that mesenchymal stem cells, endogenous mesenchymal stem cells, are largely localized in quiescent form in the bone marrow. They're tethered there based on the local SDF1 gradient. But when kidney injury ensues, there is a change in the normal distribution of gradients such that there's a marked rise in kidney expression of SDF1. There's an associated rise in serum levels of SDF1 and a concomitant reduction in the bone marrow expression of SDF1. So when we take those three findings collectively, we can envision a situation where those cells are now encouraged not to stay within the marrow, but based on the increase in plasma levels of SDF1 to move into circulation, and then based on the very high concentration within the kidney to exit the bloodstream, move into the kidney, and take up residence for a transient period of time. And in the AlloCure laboratory, we were able to demonstrate that those three things are happening simultaneously in experimental models of acute kidney injury. The most important experiments that were done to justify the successful IND submission for AC-607 was to look at animal models of acute kidney injury. The classic model is to induce ischemia reperfusion in a rat by clamping the renal artery. And following the induction of ischemia reperfusion injury, to look at the impact of pre-treating animals with a bolus of exogenous rat mesenchymal stem cells to see if we could attenuate both the magnitude of injury and the duration of injury. So in these essential experiments, the AlloCure scientists were able to demonstrate that pre-treating rats with a bolus of exogenous MSCs and then exposing them to the challenge of ischemia reperfusion injury could attenuate the burden of disease. So on the left, we see a histomicrograph of the kidney of those animals that were treated with vehicle. And this is a classic micrograph of acute tubular necrosis. You see dilated tubules, plenty of interstitial inflammation. Within those tubules, there's a setup for casts in the urine, which is a hallmark of tubular injury. On the right, we see preserved renal architecture. And this was taken from an animal that had been pretreated with mesenchymal stem cells at the time that the ischemia reperfusion injury was induced. Similarly, if we looked at a composite injury score that included both the histology as well as the biochemical measures of kidney function, we can see that there was about a 50% reduction in the attenuation of injury in these animals. We continued, based on these experiments, to look at what happened if you waited for the injury to be manifest. So now we move from a preclinical model of prevention of acute kidney injury to a preclinical model of treatment. And similar with these results, we've been able to demonstrate that 24, even 48 hours after you have biochemical evidence of acute kidney injury, treatment with administration of exogenous MSCs has about the same magnitude benefit in terms of reducing the peak serum creatinine and the duration of time that's required before creatinine comes back down to baseline. So based on those preclinical findings, the company was able to file an IND, secured Series A financing, and moved into the clinic with a phase 1 clinical trial. In that study, 16 subjects who were scheduled for elective CABG and or valve replacement and were deemed to be at high risk for acute kidney injury were enrolled and were treated with AC-607. They had a pediatric catheter threaded into their femoral artery. The tip of the catheter was visualized above the bifurcation of the renal arteries, and they had the cells administered as they came off cardiopulmonary bypass. They were followed primarily for safety, but we also took the opportunity to measure a variety of outcomes that are relevant to acute kidney injury, including measures of their kidney function using serum creatinine, their incidence of needing dialysis, and their in-hospital mortality and length of stay, rates of hospital readmission. We've now completed 3 years of follow-up on those patients with a very sound safety profile. There were no treatment-related SAEs, and the overall adverse event profile was entirely what you would expect to see in a population of patients who were coming out of the OR from CABG or valve replacement. We also took an opportunity to build a cohort of rather contemporaneously identified historical controls from the same institution where the phase 1 study was done. This would allow us to have an early look at potential efficacy with all the caveats of using historical control information. And I'll share that information with you. So the phase 1 subjects are in blue bars, match control subjects who were matched on their age, their degree of kidney disease, the kind of heart surgery that they had, the duration of time they were on cardiopulmonary bypass, etc. We identified 64 matched patients to go with that cohort of 16 phase 1 subjects. And first we looked at the incidence of acute kidney injury that happened in the phase 1 subjects versus those 64 controls. And what we saw is using two different measures of kidney injury, one on the left called a rifle, one to the right of that called ACT-AKI. Using both of those definitions, we saw a much more marked incidence of acute kidney injury in those patients who came from the control group than those phase 1 subjects who were treated with AC-607. In addition, we looked at rates of hospital readmission and duration of hospital length of stay. And similarly, as we saw with kidney function, we saw that there was an increased rate of both hospital readmission in the control group as well as a longer stay in the hospital before they had their initial discharge. With all the caveats of historical control analysis, we couldn't have asked to see anything better from the phase 1 trial, and this gave us the opportunity to go out, design a robust phase 2 trial, raise the capital to finance that activity with a Series B, and to move forward with the clinical program. So this is ACT-AKI. It's quite a large phase 2 trial looking for proof of principle. In this trial, we are enrolling patients who now have completed CABG or valve replacement and who within 48 hours of leaving the operating room have laboratory evidence of acute kidney injury with a prompt rise in their serum creatinine. At that point, they're randomized to either receive AC-607 or placebo in 1-to-1 fashion. We are measuring relevant endpoints for outcomes, primarily time to kidney recovery, then the composite of the incidence of death and or the need for acute dialysis when they're in the hospital. We'll measure their length of stay, their rates of readmission, their time in the ICU, and safety endpoints. We're now more than 50% of the way enrolled. This trial is being conducted in about 30 centers in the United States and Canada. So this is my concluding slide, so I'm getting us back on track for time. So in summary, acute kidney injury is an all too common condition. As a nephrologist, I can tell you that the community focused on renal medicine has been working at this disease for decades without a lot of success. And the concept of potentially using a local polypharmacy effect to interdict against multiple pathways and not just a single biochemical pathway is very attractive to kidney scientists. With that great unmet medical need comes the reality of a large commercial opportunity. And certainly as we've looked at this, we think that the prospect of developing the first therapy for acute kidney injury would be an enormous opportunity. Our trial is ongoing. We look forward to having the results next year, and we're encouraged by the positive feedback we've had from the regulators, our investigators, and the larger community. I thank you for your attention, and I'll certainly be here for the rest of the meeting and welcome your engagement. Thanks very much.